Good afternoon. So welcome to the stream. Welcome to the session with 26 nanoparticles and the immune system. And it's our pleasure to be here. My co-chair, Professor Grabbe, Stefan Grabbe, and myself, Volker Mailänder. We are both here in Mainz. And part of that, what we are going to present you, is part of what we have been doing in a joint collaborative research group here on nanomedicine here in Mainz. And of course, we have also external speakers here for this session. And so this is the collaborative research group 166 here in Mainz. And I know Mr. Mochini for a long time. At least we did not talk too much in Basel till now, but we have been always seeing each other. And so it was a specific pleasure to introduce Moin Mochini. He's professor of pharmaceutics and nanomedicine in the School of Pharmacy in Newcastle in the UK. And he will tell us today something about the complement modulation of nanomedicine performance in health and disease. And I think this is an important thing because the complement system is an important system in the blood system, and we will see that it's doing a lot with also our nanoparticles. So we are keen to hear what you are doing there and what you have found out. Please, Moin, please start.
Thank you very much. I hope that you can hear me and can see the presentation.
Perfect.
Excellent. So first of all, I would like to thank the chairs, the German Research Council, as well as BET for organizing this important session. And it's a great pleasure to be speaking with you on this. And I pity that we cannot meet in Basel. So what I would like to talk about is the role of the complement, and especially in nanomedicine. And starting by, so I think this is on the first slide, it shows a kind of a picture of how nanoparticles can trigger complement activation through 3 different pathways. And as you know, complement is the first line of the innate immunity, which intercepts foreign particles in the blood. And one function of the complement system is pattern recognition. And we do have these patterns expressed on the surface of many nanoparticles. And you can see the whole process of complement activation is a kind of an enzymatic cascade initiated by recognition of pattern recognition molecules on the surface of the nanoparticles. And regardless of the pathway, which is classical, lectin, or the alternative pathway, there are 3 important ramifications of the complement activation in this enzymatic process. And that is the first, which is the conversion of this molecule called the C3, and the breakdown of this molecule into a component called C3b, and then inactive C3b that act as opsonins for tagging nanoparticles for clearance and recognition by the macrophages and other immune cells. And then also a byproduct of the C3 activation is release of anaphylatoxin C3a and later C5a. And at the end, the complement, which is fully activated, we are forming a membrane attack complex. And this membrane attack complex in soluble form can actually bind to a deposit on the surface of the cells. And both membrane attack complex and anaphylatoxins could induce pro-inflammatory reactions if the whole process of complement activation is not regulated. And that could induce a lot of problems. So we have been involved for the past 2 decades to understand how nanoparticles can actually trigger complement activation. And our focus has been mainly on materials which has pharmaceutical attributes for development of nanomedicines, such as many of these polymers like polyethylene glycol, dextran, many block copolymers, and some lipid and polymer conjugates, not only in a manner which are bound to the surfaces of nanoparticles for modification of their pharmacokinetics, but also in soluble form, and that means in solution, and also some of these agents have the ability to make supramolecular assemblies. And we have found that these type of materials in different forms can trigger complement by different mechanisms, and we have been studying these from different areas in relation to nanomedicine development. For example, many years ago, we recently studied with doxyl and pegylated liposome. We actually found that the anionic oxygen phosphate on the lipid PEG conjugate was responsible for triggering of the complement activation. And that triggered a whole set of experiments with other delivery systems. And since with PEG lipid, you have a limit how much you can actually put them into the membrane, we turned our investigation into model system like polystyrene nanoparticles, and we tried to cover the surfaces of these with different type of non-ionic block copolymers, because you can characterize them very easily and you can really pack them onto the surface, and you can study them by adsorption isotherms, by AFM, and by rheology to actually understand the polymer conformation. So the idea was how much of these polymers we need to put on the surface in order to prevent complement activation. And we have found out that at a very low concentration, you have a brush, kind of a mushroom configuration. As you add more and more polymer, you become like brushes on the surface, and you can characterize them. Although you're reducing the full extent of complement activation, you can never prevent complement activation. But we found, surprisingly, this change in the polymer conformation also shifts the activation pathway, for example, from classical pathway in this case, which was due to binding of the C1q, into a lectin pathway in the systems which you have the brushes. We also came back to the liposomes to see whether these, because the studies with those polystyrene parts were very reproducible when you also had the different amount of the polymers. But since you are limited to a range of the lipid conjugate that you could incorporate into the liposomes, we came back into the actually clinical formulation of the liposomes, and we found that there are a lot of variability even within the same manufacturer, such as doxyl and Calyx, not only in terms of physical morphology, which we try to characterize in here, how much deviation we see from sphericity. For example, you see more in here than in here, but also in terms of the total number of the liposomes in each batch. And as a result, you also see huge variation among different individuals with different batches of the same formulation. And that was quite intriguing. And again, you could see the mechanisms could actually shift from one person to another person by using a different batch. But since these pegylated liposomes have a lot of interest in cancer drug delivery, and there was a report published many years ago showing that intratumoral complement activation can promote tumor growth, we actually made some of these pegylated liposomes, and we injected these into the animals, and also a different type of nanoparticles. And we saw that the particle that actually triggered complement activation was also promoting tumor growth. And in order to show this was due to complement activation and anaphylotoxin production by C5a, which is also a chemoattractant for suppressive T cells, we found by using knockout animals which are deficient, genetically deficient technically in the C5, you could actually promote tumor growth if you inject the C5 continuously when you have the nanoparticle, or even when you don't have the nanoparticles. But with nanoparticles, you could overcome complement activation and the tumor growth if you have a knockout animal which doesn't have the receptor for the anaphylotoxin, the C5a. So that is quite important in terms of the application of this nanoparticle in terms of the tumor growth. And especially when you have systems which deliver drugs, you're actually killing cancer cells by necrosis, and some of the necrotic products actually trigger complement activation. So we have to be very careful in that sense. A lot of people have talked about the presence of antibodies in human plasma that could recognize PEG, and you do see these statements in many research papers that, individuals, maybe 20-25% of the individuals have antibodies against PEG. And we have been looking into these, and from hundreds of samples that we have measured, we could rarely find these type of antibodies, and we could not even explain that until we came across this paper by Mark Scott, which was published a while ago. And actually, it's beautifully explained in here that some of these studies that actually claiming that there are antibodies against PEG are actually artifacts, and depends on the measurement you actually do and how you measure. And when you follow the right protocols, you hardly find these antibodies in many individuals. You may find in 1 or 2, but still we don't know what are the nature of these antibodies. Are they truly against PEG, or they are antiphospholipid antibodies, or could be anti-cholesterol antibodies that are recognizing or cross-reacting with PEG. Because of these issues, we also turn into the other polymer systems, such as polyoxyzilines, which have been described by Samuel Zalewski many, many years ago, becoming long circulatory in the mouse. But when we use human plasma and human macrophages, we saw massive activation of the complement oxidation by these particles, and clearance by the macrophages and the monocytes. But in the mouse, you do not see complement activation, and you hardly see clearance by the murine macrophages. So again, that is a kind of a study to warn about in terms of translating animal studies into the human. Something which is long circulatory in animals may not necessarily become long circulatory in the human. So the complement responses could be even different. Then in a very recent study that's initiated by Dimitri Simberg, my collaborator in the University of Colorado, we actually try to look what actually happens on the surface of the nanoparticles. And here we are using kind of a dextran-coated nanoworms, iron nanoworms. And dextran is another kind of a component which is widely used for stabilization of the nanoparticles. And in here it was very interesting that the opsonization process is not directed towards dextran itself, but technically once the complement was activated, the C3b was binding to proteins which are intercalated into the dextran shell of this nanoparticle. And that was a continuous process. They were binding and then they were releasing from the surface. So it was a nice continuous process in terms of the surface opsonization. We came back again recently into polystyrene nanoparticles to see, because then here you have a hard surface rather than having polymers protruding from the surface. And when you dip these nanoparticles rapidly into human plasma, this is undiluted human plasma in the presence of an anticoagulant that doesn't interfere with complement activation, you don't really see protein corona. What you see on the surface, there are different patches. And especially when you look on the screen, I don't know whether you can actually see it, you can see that the protein fibers are actually protruding from the surface. So we are thinking that maybe some of these fibers at some locations could be the nucleation point for complement activation, and we are looking into that at present. In a later paper by Dimitri, we also showed that the process of the opsonization is triggered by attachment of perhaps 1 or 2 antibody molecules to the surface of the nanoparticles, acting again as a nucleation site to form a convertase, and the convertase is attaching more C3b proteins, C3b to proteins which are intercalated in the dextran shell, which is again continuously forming and dissolving from the surface. So in order to prevent complement activation, we thought that we need to really suppress this protein intercalation into the chain. And again, in a very recent paper published this year with Paolo De Cusi in Italy, we showed that by PEG pairing, using a combination of long PEG chain and smaller PEG chains, we can really limit intercalation of these proteins, and that was overcoming, again, complement activation. And there are beautiful movies, simulation movies by Martina here, that you can go online and see it, which actually shows that this PEG pairing can reduce the approach of the protein, such as albumin, to the pristine surface once you have this PEG pairing, which again was affecting how complement convertases are actually formed on the surface. And recently, again, by understanding of the mode of complement activation, we have designed lipid systems, again, by using non-ionic lipid polymer conjugates. We have no charge, and again, by different type of screening, we showed that the lipid component can also modulate the mode of presentation of the PEG. So if you use a right lipid component, again, you could overcome complement activation. And another way would be to learn from the surface of the microbial, such as many pathogens that recruit factor H. And factor H binds to the sialic acid, carboxylic acid, and a hydroxy part. And again, by using polymers like citram, which have this type of moieties together with the right lipids, you totally overcome complement activation in human plasma. And we also extended that to design of peptide nanoparticles, which again, these are a kind of a new system that crosses the blood-brain barrier within 10 to 15 minutes, targets neurons and microglial cells, and again, show no signs of complement activation and pro-inflammatory reactions, again, by through kind of a computational design. In the last couple of minutes, I'd like to spend some time to explain some of the problems that we see in the clinic in terms of the infusion reaction to all these type of nanomedicines, as well as bigger particles in micron size, which are used in diagnostic imaging. And as you know, that there is a hypothesis called CARPA, complement activation related pseudoallergy, and we have been questioning the validity of this hypothesis. And a lot of people actually show, I mean, there are groups suggesting that the pig could be a very interesting model of simulating the human conditions, reactions, and complement could be involved. Actually, we question this hypothesis because pig, unlike humans, they have these macrophages sitting in the microvasculatures in the lung. And it has been shown many years ago by Joseph Brain at Harvard Medical School that when these macrophages ingest particles, they rapidly secrete thromboxane B2, as well as many pro-inflammatory mediators, which could explain some of these anaphylactic reactions that we see in the human. So that suggests that pig may not be a right model, and these macrophages actually playing a very key role, at least in the pig model. So to show that, we use nanoparticles, and such as polystyrene in this case, and from the same material we made different shapes such as rods or disc, and with the same volumes. And you can see that when you're presenting them to the macrophages in a static condition, within the first minute, these two type of particles are cleared less than the spheres, but later there is no difference. So we thought that because the rate of presentation of these are slower to the macrophages, maybe they could overcome the reactions, at least in the pig model. So you can see when you are injecting into the pig, the rods and these are cleared slower within the first couple of minutes than the spheres. And these anaphylactic reactions happens usually within the first minute. And when you look at, for example, the pulmonary arterial pressure and systemic arterial pressure changes, you can see with spheres it's very high, whereas quite negligible with the disc and the rods, and the same with the systemic arterial pressure, and there is no thromboxane release. So technically, if you think about the probability of presentation of a rod and a disc compared to a sphere, which is symmetrical, you are producing or you are presenting the particles at a much slower rate to the macrophages, which these reactions are overcome. If you destroy those macrophages, you can see the responses totally disappear, and doesn't matter what you inject, whether it's a pegylated liposome or a sphere or even zymogen. You can also do the slow presentation of the spheres by technically adsorbing the spheres to the red blood cells, and all of this system all activating complement. You can see again through this adsorption, the pulmonary arterial pressure actually drops, and it is a fraction of the normal sphere. And the reason why we have a small amount in here is because about 20% of the particles are still in a free mode. So in the last couple of slides, just showing that a kind of a particle volume or a equivalent surface area effect. So if you're injecting large particles into the pig, you can see a massive response. But if you inject smaller particles with an equivalent surface area to the doses of larger particles, in a sense, you are injecting more particles with the 200 nanometer size compared to the 750 nanometer size. But you can see there is hardly any reaction. So that shows that the rate of recognition by the macrophages is extremely important, and also what receptors participating in that, which actually sending the signal to the macrophages in order to secrete the pro-inflammatory reactions. Recently, we have isolated the pulmonary intravascular macrophages, and we try to see the responses in vitro in terms of the complement and ingestion and the thromboxane release. So when you have these pig macrophages with isologous serum, that is the serum from exactly the same animal, you can see uptake of the by way of comparing the uptake of polystyrene particles, pegylated liposome, and the zymogen. So you can see, regardless of the nature of the particle, you may have different extent of uptake in the presence of serum, but immediately after ingestion, these macrophages secreting thromboxane B2. When you have heat-inactivated serum, which you destroy complement system, you will see that uptake still happening to some extent. It could be through different receptors, but there is no change in terms of thromboxane B2 release. And again, that is true with the zymosan and with different type of nanoparticle, again, proving that the concept of complement activation may be very minor or may not even play a role, at least with nanomedicine. The complement is important in kind of septic shock, which you're actually producing huge amount of anaphylatoxin. With nanoparticles, you are not really producing that amount of anaphylatoxin, and usually equivalent to about 5 to 8% of what zymosan does. So we have to be very careful in that regard. So I think if the home take message with the infusion reaction is, let's not reinvent the wheel, but make it better, and not really blame complement and using inappropriate models such as pig, which may not be really relevant to the human case, and actually slowing down the production or research in clinical nanomedicine. So with that, I'd like to finally acknowledge many of my students and postdocs and many key collaborators such as Dimitri Simberg, Anand Yagmor, and Tom Moles from different institutes and various funding. So I'm very happy to answer your questions. Thank you.
Thanks, Moeen. So I don't think that I saw questions. No, there are no questions in the chat function here. So,
but maybe I have a question that I can ask you. Do you know what the exact role or what the cause of the tumor promoting effect of complement in your experiments was?
Yes, we are looking into that, and there have been many studies. So when you're triggering complement in the tumor, you are producing C5a, you are generating C5a. So C5a is a chemoattractant molecule. So what actually happened with the C5a, you see an influx of suppressing tumors, suppressing immune cells, suppressing T cells, actually homing to the tumor. So it's technically bringing the immunity down in the tumor, allowing the tumor to grow. But also recently, there has been a study coming from another group published in the Journal of Immunology, showing that the C5a is also a chemoattractant to progenitor cells from bone marrow that home to the tumor and promote angiogenesis.
Okay. One other question. I mean, complement on the surface of a nanoparticle is not necessarily something bad, and you showed examples for that also. We have shown that it can actually help to redirect the nanoparticles to certain immune cells that take them up via complement receptors as well. Is that something that you find important, or is it a sort of a bystander effect in as far as your opinion is concerned?
Well, it is a part of a defense mechanism. So the role of the complement is to opsonize pathogens, and in this case we have nanoparticles, and to facilitate their recognition and clearance by the macrophages for disruption. So it is a natural defense mechanism. So I don't think there is harm to that, but in terms of the infusion reactions, if macrophages are really involved, it doesn't have to be a complement receptor.
Yeah, okay. All right, I see no further question at the moment, but I would like to remind everybody that there will be ample time to ask questions and to discuss at the end of the session. And so it's my pleasure now to introduce my friend and colleague Volker Mailänder, here from the Department of Dermatology in Mainz. Volker is professor of nanomedicine here at our department, but he's also a clinician taking care of skin lymphoma patients. And but he's, of course, in the field well known for his work on the protein corona, as well as on endocytosis pathways of nanoparticles. And Volker, it's my pleasure to introduce you here, and please go on and give us a lecture.
So, thanks a lot for the nice introduction. The role of the protein corona in nanoparticle uptake in immune cells, that is what we and I have been working on in the last, let's say, nearly 10 years, at least immune cells. And the protein corona, that came up, of course, with a lot of help and a lot of also good discussions with Dawson Group. Why should we care for proteins in nanocarriers? And I think Muing just showed that this is an important question. Well, there's another thing to it. We attach them deliberately in order to target or to functionalize, in order to have an antigen or to have an adjuvant on the surface. And on the other hand, they attach undesirably. Whenever we put nanoparticles in contact with fluids which contain proteins, there will be some interaction. And at least if we put something like an antibody on it, there will be always an interacting part of the antibody, either the desired ones, the FAB part, the antigen binding part, or the FC part of such an antibody will do something. And in the end, it has been a problem. It's hard to predict which proteins attach, and I think this is still a problem. But we can at least guide some, we can say some, okay, this is the material, and these are the probably then found proteins now. This is the most, for me, in my personal view, the most important paper on that, what Salvati and Dawson have shown, that if you have protein absorption, then you have a cover-up, you have a mistargeting, you have a different or skewed cellular uptake, and you have a different biodistribution even on the macroscopic level. Of course, we will never have a body which has no proteins, but if you have different proteins, or if you put proteins differently beforehand on it, then you can change the biodistribution. So I just want to give you two things because of the time frame, which we have been working on. The one is the PEC stealth effect, and the other one is the targeting of absorbed nanobodies. Let me start with the PEC. So if you have always these proteins on the surfaces and we really don't want that, maybe it's a good way to get rid of all these proteins. So do this stealth effect. And there has been, of course, the idea that this unspecific cell uptake, if we put some stealth polymers on it, that we then have no proteins on it. And the significant or the most used polymer here is polyethylene glycol, and Moin just introduced that and showed that very nicely. And the idea mostly was there is no absorption of proteins. And you will see in the end, me and also other groups have now shown that there are proteins which go into the back chains. And this is dependent, as shown before, that this is also dependent on how these pack chains are packed together. And the idea was if you don't have proteins, you don't have something that could be taken up. What we usually do is we incubate our cells in plasma. We sometimes also incubate them in mice. That shows a totally different protein composition in the end. We let them absorb. This happens very fast. We do a hard centrifugation or a not so hard centrifugation. We look for cell uptake and we correlate that to these desorbed proteins, look first by SDS page and then by a proteomics approach by an LCMS quantification of the single proteins, and can detect anything between 50 to 200 proteins on these surfaces. But that's even not a capacity of the LCMS. There can be several thousand proteins detected and quantified in one run. So when we look at what's absorbed, and this has been done for many decades before, for especially also for polyethylene glycol, then a lot of people say, okay, if we functionalize such a surface with polyethylene glycol, we find there is substantially less proteins absorbed. Or you could some say, okay, that's just, that's nothing anymore. We could also say, well, with better getting methods, there are proteins, and we cannot completely hinder that. And let's take that seriously, what we find there, because it's above the threshold what we could detect. And let's look what it is. But first, the question: this is something like the experiment which we did several times because I couldn't believe it. Of course, what you see here in this case polyethylene glycol and polyethylene phosphate, which was investigated by us for an alternative to PEC. And when we put this into these nanoparticles into a cell culture which contains plasma, then you see the effect. There is very much less uptake with the functionalized surfaces compared to the aminofunctionalized surface. But interestingly, because we can do that in the cell culture, that we don't have a cell culture, at least for a few minutes or for a few hours, without plasma, then still there is uptake. Although we have the same surfaces, or we think we have the same surfaces, but we lack the plasma proteins. So is that concept really right? Do we maybe need even proteins in order to have that stealth effect? And this just shows you that you can also see that these particles are really inside without the plasma. And but when we add plasma, then the uptake is the green dots are no longer inside the cell, so the nanoparticles are no longer taken up really. And what is that? To cut a long story short, that took us something like 2 years in order really to get the story together. It is clustering that is absorbed, and interestingly, these polyethylene phosphate surfaces, which you see in the last two columns, the protein composition, that has something like up to 80% of clustering, which is an upper lipoprotein actually, and this is then obviously then doing the trick, and we've shown that in other papers, and of course because this is a Nature Nanotech paper, we need to show that in a lot of other experiments until we got that through. So the idea is no longer that we have no proteins on the surface, but we have a very specific protein composition up to, in this case, one protein, which is predominantly there. This may be also done by other proteins, actually. And then we don't have a lot of cell uptake anymore. We can suppress cell uptake very, very efficiently with these cells. And if we do the right calculation, we always see this protein composition. So how much stealth you have, you can basically determine by your protein composition in the end. Let me come to the second part. Well, we were asking how can we do a targeting now that we have something like a stealth effect, that we can avoid the unspecific uptake. And here the idea was, well, we do the chemical coupling. And this was done, at least at these days, when we did that, with ADC NHS. And it just happens that there's a buffer involved, Mainz buffer, which has a pH of 6.1. And when we do that, when we, of course, then we were binding an antibody which was directed against CD63 because we wanted to target cells that are CD63 positive for some reasons in order to get some intracellular trafficking in there. And we also were questioning if that is still then doing the targeting effect if we put a human serum to it. And if we use these covalently bound antibodies, then we see that it works, but it's very much less effective. You see the drop there in these three bars, when we add more and more human serum in this case. And you can also show that with human plasma, so that's the same effect. So and then somebody came up with the idea, actually it was Manuel Tonigold together with chemist, and said, well, we do the control experiment by just using no EDC NHS, but we just use the same buffer. And usually we wouldn't have done that because usually we would do that at 7 or 7.5. And then we just saw that first the targeting itself is better, and it drops less if you use even a lot of human serum. So absorbed something that was not surprising to us and to a lot of other people. If we're doing that all the time with ELISAs and with linked immunosoluble assays. That's exactly what we do when we do pre-code a 96 well plate there. And why is that? Well, it's because of the pH, and you see we were lucky in this case because pH 6.1 is really doing the trick, while pH 7.1 would have absorbed much less. This depends a little bit on the type of IgG you use. It depends definitely on the surface of the nanoparticle. The left two ones are carboxylated, and the right ones are amine functionalized. So this must be taken into account, but we were always able to find a pH where it works in the end. And why is it like that? Why reason why it is like that is that in this case, what you see here is you want to bind the two green parts of this antibody. These are the FAB parts, the antigen binding parts. You want to have them binding to your antigen, which is shown on the lower part, also in green, inserted into the lipid membrane of the cell. But if you're very unlucky, then you bind at least a certain proportion of your antibodies directly to the surface with the FAB parts, and you stick out the FC parts. And the FC parts is something that activates complement, that is recognized by itself, by macrophages. So that's something you don't want to do. If you're a little bit more, if you also find conformations, of course, where you only find one FAB part attached to your particle, and the other one is still sticking out and being able to interact with your surface of the cell. And of course, that would be the best, that you have the FC part bound to the surface and then stick out the two FAB antigen binding parts. And why is that working better for if you use a lower pH? If you just use the pH of 6.1 and let it sit there for a while? Well, that has been actually shown about something like 25 years ago, where they show that the FC part denatures at a low pH, and then this denatured and hydrophobic amino acids, they look for another hydrophobic surface. And that's in this case here, the nanoparticle, because that's a very large surface area, obviously. So what can we take home from that? Antibodies can be absorbed, and they can withstand other proteins when they come along. This absorption is pH dependent, and we can do that for a lot of other nanoparticles. Polystyrene, polyethyl starch, we have done that even for liposomes. In some cases you can do that, because you can also have charges on them. What can we do further? What have we done further since then? Well, definitely lipids play a role, especially when you have apolipoproteins which absorb. We have shown that sugar moieties in proteins play a role. We've also seen that the composition of your proteins source which you use. So if you allow IgG, for example, then that is doing something. There's actually in the parallel session, there is a doctoral student of mine, Adelina Haller. She's presenting together with Carol Champagne-Hack data on what heparin can do as an anticoagulant. There's a material-dependent protein corona, and we have even the influence of washing. And I think this is going to be interesting if you look at a lot of papers that have not found proteins. Because I think that is something that we need to talk very clearly about, how we wash. And with all this protruding, what Moeen just showed, proteins, I think they can be also removed by a very intense washing. And of course, we can use surface functionalization in order to attract specific proteins themselves. With that, I would like to thank all the PhD students, the co-workers, and everybody working in the lab for this wonderful, nice work that we did, not only on our own, but also with a lot of other cooperating partners. Thank you.
Thank you, Volker. Very nice talk, and it is open for questions. I still do not see any questions from the general audience. Anybody from the faculty here of the session who wants to ask a question? If that is not the case, then maybe I'll ask a question. Volker, you have been working a lot with the protein corona. Do you think that to really control all aspects of the protein corona will be necessary for a therapeutic application of nanoparticle drugs?
That we control that? Well, yeah. that we at least know what it is. I think that is easily doable in these days. That's just something that's a very, very fast analysis which we can do these days. So there should be no reason why we shouldn't be able to know that. And we can do that even in animals. So can get the particles, a lot of cases, again, out from a bloodstream, or we can do that in other models. So I think that is something that should always be known. And I think that most of the targeting, why the targeting didn't work, is because we didn't know what we were looking for in the end, or what we should avoid.
Do you think that the size of the corona proteins matters? For example, if you have an antibody that sticks out, it's a large molecule, that it will have a chance to reach its receptor despite a protein corona covering the rest of the surface? Or
I definitely think because we were also working with nanobodies, which are much smaller, and I think it will be important that we put them on large linker molecules so that we stick them out, because we will, in a lot of cases, we will still have proteins attached to the surface.
Yeah.
Or we attach something like a protein like moiety, so there are no other proteins on top of that. But that's really, really hard. And especially, as also shown before, once you have something like an FC part to it, you find other proteins which bind to biologically. It's a biological function to this one. And for example, if you look for heparin, which when you have a heparinized patient, you will have also antithrombin free on the surface of these, just because you have these other charged molecule on it.
All right, I guess we move on, don't we?
Yep. So with this, I would like to introduce Tobias Bock. Tobias Bock is a professor here in Mainz for immunology. He's the head of the Institute for Immunology, professor for molecular biology, molecular immunology. And so he's also a part of our collaborative research center. And together we have, he's coming directly from this, from the immunological side of the projects. So we would like to hear your talk. Tobias, please go ahead. Stage is yours.
Yeah, Volker, well, thank you very much. I hope you can see my presentation now. I think so.
Yep.
As Volker already pointed out, I'm more of an immunologist or molecular immunologist, but I very much enjoyed listening to all of the talks at this very nice conference, learning more and more about nanoparticles and how to use them. But before I would like to go into medias res and to talk about the tumor microenvironment and how we think in our consortium with chemists try to modulate that in order to modulate macrophage polarization, I would like to quickly thank the organizers for giving me the opportunity at all to present our data. And as a short introduction, I would like to remind you, that's probably also my immunological side, I'd like to remind you that in the immune system, context is everything. And that's so important to emphasize, because at the moment, we mainly have drugs in our hands that are clinically approved and that are given systemically. And I think there is also a great power, at least for me being a non-chemist, a great power to really understand the complexity of the immune system in a context and tissue-dependent manner, and also to employ nanoparticles to probably one day specifically target in a context and tissue-specific manner immune cells. And if we're talking about the tumor microenvironments, it's pretty much the same as if it is in any other environment. The immune cells, and it's so important, are strongly affected by the microenvironment they are working in, they are living in it, especially in the tumor microenvironment. It's so crucial, so that we think it's of utmost importance to really understand the tumor microenvironment for successful immunotherapy. And especially this is what my group is focusing on for more than a decade now, to understand this tumor microenvironment, but also context and tissue-specific immunity by isolating immune cells from different sites of the body, and especially also from tumors, but not only looking on the immune cells and thereby neglecting the other cells that contribute to the tumor microenvironment, but further to also isolate tumor cells and to understand their proteome, their transcriptome, their secretome, all the factors that may influence the immune cells, talking to the immune cells to act in one or the other way. And by doing so, a very talented postdoc in my lab, Toshka Bohm, got interested into something that is due to the high metabolic demand of cancer cells, which is this aggressive growth that you know from cancers. And at the same time, probably because of this aggressive growth, there's less angiogenicity, resulting in less blood vessels. And with that, in a very reduced buffer effect. And Volker already referred to you about the importance of the pH and chemical reactions. And I would very much like to emphasize in my talk on the effect of the pH value on the immune response. And Toshka get very interested in this acidification, at least in certain areas of the tumor, in this acidification process. But to really have something in hand to study this acidification, she needed to find tumors that, if you like, represent extremes of high acidification and low acidification, just to compare the different tumors, and not comparing tumors with non-cancerous tissues, for example, and then measuring, if you like, effects that are not specific for tumors. So to understand that, she teamed up with a very talented bioinformatician that was at that time point working at the molecular genetics department here in Mainz, looking into already published data sets, tons of data sets, like the TCGA, the Cancer Genome Atlas, and other atlases, to look for gene expression in different tumor types that are associated with a high metabolic demand. So, for example, high glycolytic activity, which then we thought might resemble also the potency for high acidification. And by doing so, Toshka and Steffen realized that especially melanomas, aggressive melanomas, are not so aggressive in that case, at least when we studied them. Colonadenocarcinomas seem to display extremes of glycolytic activity, at least with respect to the expression of genes that are important in glycolysis, and therefore, as I said, may display extremes in that case. But really, how to measure acidification in vivo? And there are some techniques to do that, but to do that on a global level, if you like, on patients that are probably not even here in Mainz, we had to find another possibility. And we were struggling with that until the publication by Bob Gillis' lab came out, showing that expression of LAMP2 seems to be associated with acidification. So we went back not only into mouse models, but also into human data sets that were already published, searching for LAMP2 expression. And indeed, our suspicion that melanomas, being quite aggressive, at least the ones we studied, seem to display extremes of high glycolytic activity. When we look into the humans, we also see higher LAMP2 expression, we think, as a surrogate of acidification in these melanomas when compared to colonadenocarcinomas, and vice versa. If you look into the mouse models, into the Black 6 mouse, the B16F10 that a lot of people refer to already, and the colon adenocarcinoma in the very same genetic background, which is called MC38. So mouse and human melanomas, or SKCM, show comparatively high acidification. At least that was what we concluded from this LAMP2 expression data. But how can tumor metabolism, and especially acidification, influence now the quality of an immune response? And to understand that, we did different experiments. We cultured different immune cells under different acidic pH conditions. But again, we went back into bioinformatic analysis to understand that on a global level. And there it helped that other people were already quite intensively working in that field, people that were probably also working with plants. But the beauty of nature is also something that if something is really good, it's conserved. So it seems as if there are G protein coupled receptors expressed not only in plants, but also even in human beings that are capable of sensing protons. And usually these G protein coupled receptors are of the G alpha S, so the stimulating phenotype, stimulating the de novo production of cyclic AMP by ATP, which gives rise then to downstream signaling. But what is the cell type that is most attracted or most influenced by protons, or by sensing acidification, in our case, in the tumor microenvironment? So we did some in silico analysis of already published data sets and found out that especially macrophages, cells that you've probably heard of, already this morning for quite some time in different talks in this cleanup Congress, seem to be well equipped, compared at least to other immune cells infiltrating the tumor, seem to be well equipped with, especially with these proton sensing G protein coupled receptors. So with that result in hand, our attention was mainly on the macrophage in our project. And we tried to, first of all, in vitro, but also ex vivo, try to understand how specifically acidification affects the macrophage in its function. So we isolated macrophages from mouse and human melanomas and conducted deep sequencing, RNA sequencing experiments. And we compared these data sets to macrophages ex vivo that were either cultured in organic acids, so to understand whether there is, if you like, a direct effect of lactic acid, which is uncoupled to the low pH effect that's given rise by acidification, or we used inorganic acids to just adjust the pH that we can measure in a tumor now. And then we compared these different data sets, and you can clearly see that melanomas themselves seem to induce a specific transcriptional program in macrophages. Acidification does so, and also lactic acid does so. But there seems to be a common denominator within all these different analyses, at least on the effect of the transcriptome of the macrophage when it's under acidic pH conditions. And within this common set of genes that are regulated in melanomas under organic acids or under the influence, just if you like, of acidic pH adjusted by inorganic acids, you do see that there are genes where some people refer to that they are markers for, if you like, M2-like macrophages, anti-inflammatory tissue repair-like macrophages, also markers that gave even rise, the identification gave even rise a few years ago to the Nobel Prize, so the HEF1 alpha that can modulate function under hypoxic conditions of macrophages. But we also found a molecule which caught our interest, which is called ICER. It's a transcription factor called inducible cyclic AMP early repressor. And if you remember how these G protein coupled receptors signal via cyclic AMP, we were very much interested in that transcription factor to understand how this transcription factor contributes to sensing not only of acidification, but also to the functional consequence of this acidification on macrophage polarization. And to really understand the role of this transcription factor, we did something that usually immunologists, at least molecular immunologists, mouse immunologists usually do. We generated a conditional knockout mouse by LOXP flanking an intrinsic promoter that's not detailed within the CRAM locus that's solely responsible for the expression of this transcriptional repressor. We LOXP flank this locus within the CRAM locus and then cross these mice that we generated on the black 6 background to mice that express CRE under the LYZ2 gene, often referred to as lysine CRE, mice which give then CRE expression in macrophages and mainly myeloid cells, resulting in mice in the offspring of these mice, resulting in mice that carry the LOXP flank allele and the CRE expression in myeloid cells, so lacking ICER in myeloid cells. And then we wanted to know what really is the impact of ICER deficiency in myeloid cells, mainly macrophages, but also granulocytes, I have to admit, in these different tumor models. So in these low glycolytic, probably low acidifying, and high glycolytic, high acidifying tumor models in these mice. So we inoculated glutamate control mice as well as the mice lacking ICER in the myeloid compartment at the same time with melanomas and colon adenocarcinomas to really have the controls, if you like, on the very same animal. And we were happy to see that the low glycolytic, non-acidifying MC38 was not differently controlled when myeloid cells lack ICER. However, if we look on the high glycolytic tumors, we do see that there is a very efficient immunity induced and very efficient anti-tumor immunity, I have to say, induced in these high glycolytic, most probably high acidifying melanomas when myeloid cells are here in reddish, when myeloid cells lack this transcriptional repressor. So we concluded that myeloid-specific deficiency in ICERs results in robust anti-melanoma immune responses. But what's going on here on the molecular level? You learned that I'm a molecular immunologist, so I want to know things in detail, especially down to the transcriptional level. So what did we do? We isolated the macrophages from these tumors, the invading macrophages, which then have a ISO-deficient genotype or ISO-competent phenotype in the case of the glutamate, and then performed again unbiased single cell RNA Sac sequencing. And if you just look on the bulk Sac analysis of these data sets, you do see something with I found very intriguing, because if you compare these high glycolytic, high acidifying tumors with the low glycolytic, low acidifying tumors, you do see that the same genetic deficiency in ICER results in completely different outcomes, emphasizing the role of the different tumor microenvironments and how specific we probably have to treat tumors, how to, personalized, if you like, we have to apply immunotherapies here. And if you take a deeper look into that and just look for pro-inflammatory genes, often referred to as M1 associated genes, like these pro-inflammatory cytokines, you do see that in the absence of ICER in B16 melanomas, all these genes are upregulated, and in the presence of ICER, they are downregulated, whereas there's diverse regulation of these genes in the MC38, in the absence of ICER, as well as in the presence of ICER. But in the wild type or glutamate control mice, you do see that in the presence of ICER, genes associated with an M2 or tissue repair phenotype, alternatively activated macrophage phenotype, often referred to, are upregulated, and these genes are downregulated when ICER is lacking. So myeloid-specific deficiency in ICER not only results in this enhanced anti-melanoma immunity, but it seems as if it renders a non-inflammatory tissue repair type macrophage into a pro-inflammatory macrophage, especially under acidic pH environmental conditions. But now to bring in the nanoparticle part, if you like, of my presentation, we had to understand how to employ that therapeutically, this knowledge. And there are several stages where you could interfere. You could either try to adjust the pH within the tumor. That's possible, but also a challenging task. You could try to prevent expression of ICER by employing nanoparticles, delivering small inhibitory RNAs, for example, or something else. Or you could adjust the signaling pathway at the level of the analyte cyclases. And we decided, for the sake of simplicity at least, because we had some compounds in hand that are able to modulate here, to modulate at the level of cyclic AMP generation. But to do that, we first had to establish a system where we can easily measure the effects. And we decided to do that first in vitro and to see whether or not at all we see, by adjusting the pH via anorganic acids, whether we see rising of cyclic AMP in the macrophages. And you can clearly appreciate that we do see that. We do see, based on this rising of cyclic AMP, we do see high expression of this transcriptional repressor. And if we inhibit in that in vitro setting, if we inhibit with a PON ADCY inhibitor, de novo production of cyclic AMP, we do inhibit de novo production of cyclic AMP in the macrophage in vitro after and the expression of the inducible cyclic AMP early repressor. But now then, we teamed up with colleagues from this collaborative research center that Volker was already referring to, with Matthias Barth's groups, and here especially Alex Birke took over the task to generate nanoparticles, in that case my cells. And please excuse that I'm not an expert here, so I can hardly answer questions referring to the nanoparticle itself, but I will try to if you have one. And then loading this inhibitor for analyte cyclase activity into that, and then injecting the loaded micelle into mice that carry either high glycolytic or low glycolytic tumors. So we asked the question, if you like, can we generate polymer micelles that continuously release just an ADCY inhibitor to prevent GPCR-dependent signaling, and with that, the polarization of the macrophage. So we, again, inoculated mice with either the B16F10 as a model for the high glycolytic tumors, and at the same time with the MC38, the model for the low glycolytic tumors, most presumably also low acidic. And then after palpability of the tumors, we started the treatment with these nanoparticles that were produced in Matthias Barth's labs. And then in the MC38, we didn't see much of a difference, no difference at all, either if we used the empty nanoparticles or if we used the NP-MDL, so the MDL12 loaded nanoparticles, or just the buffer that we used as a control. However, if we use the low glycolytic activity, the high glycolytic activity B16F10, and then injected the nanoparticles upon palpability, we clearly see a tremendous effect, I would call that, on the growth of these high acidic melanomas. So we concluded that indeed pharmacological inhibition of acidification induced cyclic AMP production in that case, not specifically targeting macrophages, I have to admit, but we think based on recombinase expression experiments that it's mainly the macrophage that's responding here, results in robust anti-melanoma immune responses. So taken together, I take the opportunity that others summarized what we've already published in this field by a very nice overview of our publications by Sakin and colleagues, that high glycolytic activity, not only in tumors, but also in tumors, modulates the polarization and the function of macrophages, YHSG protein coupled receptors involving cyclic AMP de novo production, expression of ICER. I didn't show you the data on pro-inflammatory cytokines, how this transcription repressor suppresses expression of these pro-inflammatory genes, thereby, we think, inhibiting a negative feedback loop on the macrophage, with then further extending expression of that and not inducing this tissue repair type macrophage phenotype. And that we think is really worth to understand the nature of the tumor microenvironment, also in case of pH values to modulate that. With that, I'm coming already at the end, to the end of my presentation, and that brings me to the acknowledgements. And I already introduced to you Toshka and the bioinformatician that was together with her in that project, Stefan Rapp. They did tremendous work together with people in Toshka's group, Natalia and Nabil, but with great help from the other lab members. And especially, I'd like to acknowledge our collaboration partner, Christian Becker, in the dermatology department, and especially Matthias Barth and Alexander Birke. At that time, they were at the organic chemistry here in Mainz, now moved to the Netherlands, but we are still nicely collaborating on these projects, trying to improve the nanoparticles, and one day hopefully even get macrophage specific targeting, one day probably also the tumor microenvironment, but that's something, that's our wish. Thank you very much for your attention. I'm happy to take questions.
Okay, so we still don't have questions from the audience.
So maybe I can step in, Toby. Very nice talk. I noticed that there was quite a significant disparity in gene expression induced in macrophages by protons, acids themselves, rather than by lactic acid. The genes that are induced by lactic acid or by lactate cannot be induced by the acidic function of lactate. Is it are those all glycolysis genes, or what is it that is induced by lactate?
You mean induced?
Yeah.
So as you can imagine, we are still working on that signaling pathway, and Toshka and Natalia now have data in hand that it's already falling between on the level of the receptor. So it seems as if GPCR65 is mainly responsible, at least in these mouse models, for sensing protons, while GPCR132 seems to be the major G protein coupled receptor being responsible for recognizing lactic acid. And if you look up these genes that are exhibiting lactic acid, that this seems to render also the metabolic activity of the macrophage. Probably also that I'm not referring to at the moment by a non-GPCR132 dependent mechanisms, but probably also by using lactic acid, if you like, and then further using that into in the metabolism of the macrophage, and that seems to somehow also in G protein coupled receptor different ways to render a gene expression in a macrophage and the polarization. Yeah.
All right. As there are no further questions at the moment, and again, I'd remind everybody that we do have questions and debate at the end of the session. It is now my pleasure to introduce Lutz Nuhn. Lutz is also here from Mainz. He's a chemist. At present, he is junior group leader at the Max Planck Institute for Polymer Research in the group of Tanja Weil. And Lutz is something that I would call a real sort of person who crosses barriers. Although he's a chemist, he understands a lot about biology, and he's using this very effectively in his research. And Lutz, I'm anxious to hear what you're going to say about polymeric nanoformulations to promote immunotherapy responses. Please go ahead.
Thank you very much for this kind introduction. Can everyone see my screen now? Yeah? Okay, perfect. Thanks also for all the organizers, especially to give us this opportunity now, especially also thanks to Bert Löffler, that he brings to the community now together with this online event that we can share our information and our research. And thanks also to Stefan for this kind introduction now. Yes, I would like to give a little bit of materials perspective in this topic of immunotherapy. In particular, I'm working on polymeric structures or polymeric nano formulations and how they are capable of promoting immunotherapy in different ways. Well, in fact, when we look at the immune system and how the immune system is interacting with pathogens or foreign materials and how they process them and also derive then immune memory response, we are often seeing how very sensitive they are interacting with especially nanometer-sized materials, be it fragments of bacteria or also nowadays the viruses, especially the SARS-CoV-2 viruses, and the massive immune responses that they can cause, and how then patients need to stay in the clinics and get treated with anti-inflammatory drugs. On the other side, we have the material sciences now, and we are nowadays have made more and more progress in synthesizing nanoparticles, or also especially polymer-based nanoparticles, which are can be very highly functionalized by, let's say, putting functionalities inside the core or decorating the surface of the material. We have heard in the first talk also about how to prevent then absorption from opsonines and pegylating materials, but also the second talk placing targeting moieties onto it. And this is also all what we are focusing on, how then these materials in particular are capable of interacting with immune cells. And this way of how immune cells can interact with nanoparticles has already been shown for a long time. And I have chosen just one example from the Hubble lab, who showed at the time when they injected different nano beads based on latex subcutaneously of various sizes, and then looked where these particles actually end up. They found them at a size below 100 nanometers, accumulating in lymph nodes and in those areas where you have a high density of immune cells. And moreover, then these materials are taken up and processed very efficiently. Well, during my postdoctoral time in the Bruno de Geest lab in Belgium, I also we continued this research, and we used core cross-linked polymer micelles, or we call them polymer nanogels, because the core was also hydrophilic. And we repeated these experiments, and with our 50 nanometer sized materials, we observed similar characteristics after subcutaneous injection, a massive accumulation of lymph nodes and uptake by immune cells. The feature of these materials is that it can also degrade under endosomal pH conditions into then single fully hydrophilic water-soluble polymers. And when we injected them, we saw that they did not accumulate in these lymph nodes and were also less internalized and processed by immune cells. So we have a kind of like a sweet spot in this nano regime between 10 and 100 meters, where we can very effectively, by a passive approach, induce an interaction with immune cells. And now coming to this chemical structure and how we fabricate this nowadays in my lab is, well, we use a classical blockopolymer approach where we synthesize these amphiphilic structures that self-assemble into polymer micelles, and then we do a post-polymerization modification step and modify the core and introduce functionalities or also crosslinks that allow us that these particles cannot fold under pH conditions, under acidic pH conditions. And coming to the chemistry, how we fabricate these blocopolymers, we use classical controlled radical polymerization techniques, but nowadays also metathesis polymerization techniques to get a more rigid backbone, or also more lately ring opening ionic polymerization strategies to introduce also degradable features in the backbone of these polymers. And when you look at these structures, then the hydrophobic domain that they are composed of are these perfluorinated pentafluorophenyl esters. And they have a nice feature because you can here do an amide formation and replacing these actually rather toxic pentafluorophenyl groups and introduce then new functionalities into the core of these micelles. And for instance, what we introduced were via these bisamines, such ketyl groups, which undergo, as already mentioned, under endosomal pH conditions, a degradation into single polymer chains. Unless or beyond introducing crosslinks, we can also introduce functional groups or also pharmaceutically active molecules. In this case, during my postdoctoral time with Bruno Duchies, we were working on TLR7/8 agonists, in particular imidazokindolines, so small molecules that can also activate these innate immune receptor that then causes maturation of antigen-presenting cells. And we could make use of such a derivative that had a primary amine, which allowed us to couple it into the core of these particles with only a minor loss of receptor activity. So we were still in the below micromolar regime of receptor activity. But this had a tremendous impact on the pharmacokinetics of this material, of course. When we injected the small molecule subcutaneously, we could see how it distributed over the whole body. And here in this immune activation reporter mouse, monitor how it immediately caused severe systemic inflammation that was reduced via this coupling to this nanoparticle, and we could really localize this immune activity only to the draining lymph nodes where the particle is traveling and is massively interacting with the local immune cells. This then had also an impact that we could, for instance, use these nanogels bound with this IMDQ as adjuvants for vaccination, also lately for antiviral vaccination. But we also observed another nice feature, as lately been addressed in the last talk by Tobias Bob, with this immune suppressive state that is present in the tumor microenvironment. We thought if we inject the particles now peritumorally into the tumor microenvironment, that we then could again see how locally we introduced a local immune response instead of the systemic inflammation. And this had also then an impact on the rather immune suppressive immune cells and also reduced the tumor growth. Moreover, we could show also in this model that we then also induced with a B16 over tumor over specific CD8 T cells, so that we believe with our local immune therapeutic that these nanogels train to the dendritic cells or the antigen-presenting cells in the draining lymph nodes of the tumor, where also antigens from dying tumors are accumulating or also arriving, and then induce this specific T-cell mediated anti-tumor response. Another approach that we are now following within the Collaborative Research Center, as already been introduced to you, is that we are now also more aiming that we cannot always treat tumors locally, especially when they are hidden far in the body, and that we have rather systemic approach that we can inject our nanogels systemically, and that they themselves can then interact with immune cells in lymphoid organs. But for this purpose, then we have to also increase the degree of functionality of these carriers, that they also deliver the antigen to the antigen presenting cells. And this is work that I'm now presenting you as done by my co-workers Judith Stickthorn and Jennifer Halbrook from Hans-Jörg Schild's lab at the immunology department in Mainz. And what Judith first had to do was increase the functionality by placing chemically orthogonal addressable groups on the surface of these particles. In this case, these were azide groups that were then addressable by click ligation techniques to couple antigens onto the surface. For instance, she has coupled an ovalbumin that was fluorescently labeled and is here just purified by or analyzed, characterized by an SDS page. And she could show that this is quantitatively coupled to these nanogels, and also in vitro, then it is co-delivered or co-taken up into antigen presenting cells together with the particles, compared to here just the physical mixture of the two components. And of course, the co-delivered immune stimulant then also had an impact on the maturation of these antigen-presenting cells. So we saw an upregulation of MHC2 and CD86 to then also look at the T-cell response that could be induced with these particles. And for this purpose, we again compared the covalent deconjugated construct with just the mixture of antigen with nanogel, where we already had good experience from our previous immunization strategies. And in fact, we saw a beneficial impact when we coupled the antigen to the particles, because then here, especially CD8 T cells, so cytotoxic T cells, were increased for the physical, for the chemical conjugate versus the physical mixture. And this was just demonstrated in vitro, but could also be proven in vivo, that we saw an increase in CD8 specific T cells against in this case, when we injected these particles intravenously and looked for these T cells in spleen. We also looked for the antibodies in the serum against the antigen that we co-delivered, and couldn't then observe any difference between the two constructs. So this not really difference between the antibodies was then also reflected when we first immunized the mice and then did a treatment with a challenge with immune cells that were expressing the ovalbumin. Then we couldn't really determine any difference between these two constructs, simply probably assume that the antibodies that are present here during the injection of the tumor cells is very strong to prevent the growth of the tumor. However, it looked a bit better when we first challenged the mice with the tumor, and then were therapeutically treating these mice with our vaccine construct. We saw that then the conjugate had a slightly benefit on reducing the tumor growth, which we suppose is caused by this increase in CD8 T cells. Moreover, we looked then also that these reduced tumor growth is really antigen specific. And for this, we repeated an experiment and challenged the mice with two different types of tumors, just the MC38 tumor model, and here the MC38 expressing ovalbumin at the membrane. And there we saw that especially those tumors that were expressing the ovalbumin were specifically recognized and showed a reduced tumor growth compared to the MC38 alone that was growing simply like untreated mice. And in these tumors then that were growing less, we also saw an increase of immune cells, mostly myeloid cells, but also a slight increase, not that much significant, but a slight increase also in CD8 T cells as shown here. So to sum up, this story allowed us now really to, with our polymeric construct and coupling also two functionalities onto this carrier to better address then also the adaptive immune response. And if I might have still a few minutes, I would like to also concentrate on another aspect that is shown lately in the last talk by Tobias Bob, playing an important role. These are these tumor-associated macrophages, where we were also interested whether we can also change their phenotype from a pro-tumoral immune suppressive phenotype with our toll-like receptor agonists into a more pro-tumoral phenotype that they can support the T cells interacting and eliminating the tumor. And for this purpose, my coworker Max Scherger teamed up with Eva Bolli from the group of Jo van Ginderachter from the Vrije Universiteit Brussels. And in the Ginderachter lab, they have developed a nanobody, so a single chain antibody fragment that is recognizing the CD206 receptor, or that is overexpressed or heavily expressed on these M2 type tumor-associated macrophages. And we could engineer them with the C-terminal cysteine that allowed us to really specifically, without damaging the internal disulfide, conjugating, for instance, here fluorescent dyes. This is a near fluorescent dye, which we could really quantitatively conjugate and got really monofunctionalized constructs that allowed us to monitor with this near-infrared dye the body distribution and really demonstrate how these nanobody is binding in the tumor. And inside the tumor, we could then also see that it is binding specifically to the CD206 or macrophage mannose receptor MMR expressing macrophages, because the knockout mice that did not where the macrophages could not express this receptor, we couldn't find any binding. So instead of coupling fluorescent dye, of course, we could then also by a chemical modification in collaboration with Paul Besenius from the University of Mainz, then also fuse toll-like receptor 7/8 agonist, our imidazoquinolone derivative, on the C-terminus of these nanobodies with a minor loss of activity. But nonetheless, this was still capable of changing the phenotype more or less of these macrophages, because when we were isolating M2 type macrophages characterized by MHC low expression, but MMR high or CD206 high expression, and then treating them with our nanobody construct, we could increase the level of TNF alpha, IL-6 and NO. So we then moved on and injected these nanobodies also into tumor-bearing mice. And we saw then here a significant reduction in tumor growth for our nanobodies that were carrying this TLR7/8 agonist. And I have to mention that this was also really nanobody specific, this reduced tumor growth, because when we injected a non-control nanobody that was not binding to the macrophages, we couldn't get any such a pronounced significant reduction in tumor growth. And when we then sorted out of these tumors the macrophages and looked again at their via NGS or by PCR, I have to say, at their expression of M2 type and M1 type genes, we saw that with the treatment of our nanobody, we saw a decrease of typical M2 type.
Looks your over time.
Yeah, I'm already finished now. This is just the two last slides that I'm now sharing with you, that these genes were decreased while the M1 type, typical M1 type genes were increased. We also saw that this promoted the infiltration of effector T cells in these tumors. So at the very last end, I just want to show you, of course, here we were only delivering one TLR7 agonist, and we are now working on decorating nanoparticles with these targeting moieties so that we can also deliver more TLR7/8 agonists into the tumor. We just done already this with this last cartoon here showing that we can already target these macrophages when we have coupled these nanobodies to the surface. We can selectively find an uptake in these tumor-associated macrophages versus knockout mice. Okay, so this is my last slide showing you the opportunities that we have with, on the one hand, the nanobody strategy to target immune suppressive immune cells in the tumor microenvironment, and with a nanogel approach, how we can install active immune responses and install an adaptive immunity. And of course, with our collaborators that we have within the Collaborative Research Center here in Mainz, we are now interested also to tackle various other aspects in the tumor microenvironment to combat tumor growth. So at the end, I would like to thank my co-workers, my team here at the Max Planck Institute, my co-workers from the Max Planck Institute, also the co-workers within the Collaborative Research Center and the partners from Belgium and the funding agency. And I'm happy to take questions now here or also later on in the Zoom session. Yeah.
Lutz, thank you for the very nice talk and the much data you shared with us. So are there any questions? We did not see, we just saw that there are a lot of questions, especially for the protein corona talk systemic or local effect of the nanogel.
Excuse me?
Thank you a lot for interesting data. It's surprising that it requires IV injection to see a system effect if the mode of action really is drainage to nearby lymph nodes and antigen presentation there. In that case, I would always expect a systemic effect after intratumor injection. Have you looked for systemic effects, second tumor, spleen, weight, also in the case of tumor injection, or have a comment on that?
Yeah, so of course, these local approaches were at the beginning quite attractive and also did not require the antigen co-delivery. That's why we started with them very early. And so the systemic approaches that we are now following, we now also check how much we influence the toxicity or the systemic inflammation. We could show already that the systemic inflammation is reduced because of our nanogels. We assume that we have a preferential accumulation in the spleen and other lymphoid organs, and this promotes our immune response.
Thanks, Lutz. So I try to see if there's another question, because we always have to upload again here the questions. It's not automatically done, but there is no other question. So I think we go on, and we will try to see all these questions that we have now in the chat. We will do right at the end for all the other talks, and have all these questions together with all the other presenters. Okay, with that, I would like to come to our last presenter today, which is Yulia Bimova. She's professor of neurosurgery and biomedical sciences, director of the Nanomedicine Research Center, and a translation program in the Cedars-Sinai Medical Center in Los Angeles. And she's going to talk about the effect of glioblastoma microenvironment on the local immune system, FDA preparation of clinically suitable nanobioconjugates. So we will see how much clinical relevant we get here. I think that's a very interesting talk in terms of translation into the clinic. Please, Yulia, the floor is yours.
Hello, it's still morning in Los Angeles, 6 o'clock in the morning. Do you see my screen?
Yes, we see your screen, but not in presentation mode.
Okay, so this is my title slide. I am going to speak about treatment of brain cancer through modulation of tumor microenvironment. And I would like to say a few words about chemistry which we use. So we use biodegradable nano polymer, which we obtain from slime mold, Plasmodium. And we have the technology to produce it in pharmaceutical grade purity, which is supposed to be 98–99%. And this is exactly what FDA requires us to do. So, I'm sorry. I don't understand. Okay. So we are constantly working on polymeric acid nanomedicines, cell membrane permeation for better delivery into the different type of tissues and of course cells. So I'm sorry, why it's jumping all the time. So we prepared a copolymer. Our polymeric acid polymer was conjugated with trileucine copolymer, and this gives us a low pH. We spoke about importance of low pH, which we have in the tumors, obtain more hydrophobic moieties, which are oligomerized and much easy permeate cell membranes. So modern cancer treatment necessitates the ability to regulate all components of tumor microenvironment. It's supposed to be expression of oncogenes, it's a regulation of stem cells, expression of immunological checkpoints, and regulation of extracellular matrix. So, overview, what is going on in glioma immunotherapy. I would like to say a few words that glioma, according to National Cancer Institute, it's only one last untreatable cancer. And this review from Nature Clinical Oncology summarizes a number of clinical trials which were done during a few recent years. It's a clinical trials for vaccines. It's a clinical trials for oncolytic viruses, immune checkpoint inhibitors, about 15 clinical trials to treat gliomas, genetically modified T cells, CAR T cells. And unfortunately, treatment of glioma, it's not such as successful like melanomas, lung cancer, few other tumors, which we now treat with checkpoint inhibitors or CAR T cells. And the summary of GBM treatment, no immunotreatment or combination so far has been superior to standard of care GBM treatment, which is temozolomide in combination with radiation therapy. This type of treatment, which was introduced 35 years ago, gave us 2.4 months improvement in treatment. And we do not move since that time to better treatment of human brain cancer. So what we wanted, we wanted to see what is able to do in terms of regulation of brain immune system as a component of glioma microenvironment. And something wrong. Yes, so this is our idea. So we know that checkpoint inhibitors as IgG1 monoclonal antibodies, they do not cross blood-brain barrier. Do you see this movie? It should be motion, yes. So IgG monoclonal antibody, anti-CTLA4 or PD-1, they do not cross blood-brain barrier. When we put them, when we covalently conjugate them to our nano platform, and as a part of nano-immunology agents, they can cross BBB through a receptor-mediated endocytosis. And then checkpoint inhibitors in the interstitium of a brain, they can block Tregs and activate cytostatic lymphocytes, or natural killers, and macrophages special fraction, and kill cancer cells. But it was our idea because treatment options are limited for the central nervous system. Very important that our nano-immunodrugs should be fully biodegradable. Then, and after the systemic injections, we can reduce the tumor size, tumor proliferation. So it was our idea, and this is the data. I'm very sorry. Yeah. Okay, so we synthesize and validate our nano-immune drugs to treat brain tumors, and we're supposed to validate the presence and activity of anti-transferrin receptor monoclonal antibody or peptides for brain delivery through endothelial system, and also after our synthesis, everything connected covalently to the polymeric acid platform. It should be vital. So our results in ELISA confirm the fact that our synthesis is not damaged activity of our antibodies or peptides. Next. Here, this is a mouse brain, and you can see only penetration of our rhodamine labeled antibodies, PD-1 or CTLA4, that they are visible inside the brain tumor. However, naked antibody, we do not see any signal in the tumor when they are not part of nano-immune conjugate drug. Here it's high magnification, and again, the naked antibody there is stuck in a blood vessel, or when they are part of nano conjugate, red color, you can see it's rhodamine, they are outside of blood vessels. So it's evidence that our construct, our chemistry goes through blood-brain barrier. And we have a quantitative confirmation as well. Here you can see that survival of our animals are much higher because we activate brain tumor local immune system, which is shielded by a blood-brain barrier, and we have better survival in combination of CTL4 and PD-1. And here we replaced monoclonal antibodies by peptides. It's AP2 peptide, and survival of our animals bearing gliomas are much better. Here it's, I'm so sorry. I'm going back. Okay, and here, this is a summary of the mechanism of local brain tumor immunostimulation, and in phase 1, we activate macrophages, fraction 1, anti-tumor macrophages, which destroy cancer cells. In a second pathway, when we use anti-PD-1 antibody, we inhibit tumor cells. I'm sorry, I don't know what is going on. Yes, and okay, and third, ADCTL4 also provide local inhibition Tregs. So very important that all these three independent pathways, they contribute for inhibition of tumor cell growth, activation of other fraction of immune cells, which produce interleukins. I did not show here our data from flow cytometry, activation of interleukins, and increasing fraction of CD8 lymphocytes. So what is important to say today, that only checkpoint inhibitors, it's not enough. We have much better treatment of brain cancer when we combine checkpoint inhibitors suppression with molecular markers and targets, endothelial growth factor receptor suppression using morpholina antisense and c-MYC. So here, tumor microenvironment, as I said initially, it's very important when all components work together. So we regulate immune system, we also suppress specific molecular targets, EGFR, we suppress angiogenesis, and c-MYC, which is known today as stem cell marker for cell proliferation, and very powerful regulator of immune system. So this is blue curve. It's a survival of our animals, which we published in Nature Communications last year, and red curve, it's survival of mice bearing brain tumors when we combine treatment of regulation of checkpoint inhibitors activities with angiogenic and another immune stem cells regulators as c-MYC. So we received 20 days difference in survival, and it's significant numbers. We speak about brain cancer treatment. Here, it's our preparation, our nano-immunodrug preparations for FDA submission, and our drug we injected into monkey in 1 therapeutic dosage and acute 10x dosage. So it was 2 male, 2 females. They were 3 injections. It's important because we wanted to see immunotoxicity. So when we finished this study, our conclusion was that no pathological abnormalities were noted in toxicity study. So we studied comprehensive panels for toxicity and PK. So we produce the drugs already not in milligrams to treat mice. We produce it in grams. So in conclusion, I hope I was able to convince you on possibility of activation of brain local immune system by delivering checkpoint inhibitors through blood-brain barrier. The importance of tumor microenvironment in glioma progressions that is largely independent of heterogeneous genetic mutations in glioma. So we do work with biomarkers. We constantly try to find new biomarkers to treat gliomas and other cancers. But we found that microenvironment is much more conservative, and regulation of tumor microenvironment maybe would be more beneficial for treatment of gliomas and some other cancers. And number 3, the role of multifunctional nanodrugs in the future tumor treatment is also essential for the better results. So a dream nanodrug, I would like to stress here, for clinical uses should be multifunctional, non-toxic, of course, non-immunogenic, targetable for inhibition of specific markers at the same specific site, and efficient for treatment. And FDA, this is our end point, which we have to cross or satisfy, requires the synthesis of all our nanomedicines should be reproducible, scale up. This is what it took us several years to achieve. Drugs should be non-toxic, molecular biological mechanisms should be fully evaluated, efficacy of treatment is demonstrated, and we are going all this now to prepare our drugs for clinical trials. Thank you very much for your attention. I am very thankful for people with whom I work so many years, in particular excellent chemist, Dr. Egerhard Holler, and many, many other team members. Thank you very much.
Thanks, Yulia. Thanks, Yulia, for that interesting talk. We have one question in the chat, which now works, as we got to know how to work with that. So the question is, do you have any ideas about retention of your nano antibody complexes inside the brain? Are they removed via the lymphatic system? And if yes, how long might that take?
Okay.
And it was Nicholas May who was asking this question.
They definitely retention because it's a high micromolecular drug. It's not a small molecular drug, but it's a fully biodegradable. I tried to demonstrate it in a movie, but we measured PK, we measured all pharmaceutical parameters which are important, and this is why we use with this natural biopolymer, which is fully degrades. We also use all components we covalently attach to this natural polymer. They are also biodegradable. So half-life of our drug, it's from 10 to 12 hours. And this is what we publish, study constantly, because we know it's crucial questions for FDA.
Thank you very much. So,
can I add a question?
Yes, sure.
Sure.
Yeah, thanks for this nice talk. I was just wondering about the mechanism of action, how your polymer is then promoting the passage through the blood-brain barrier. Is it simply hydrophobic interactions, as you at the beginning mentioned, but then it would also probably stick to fatty tissues, or what is the mechanism of action there?
Okay, so hydrophobicity is important component for blood-brain barrier delivery, but this is only the top of the iceberg. Our nano-immune drug going through BBB using receptor-mediated transcytosis. Okay, yeah. And for this, we're using either monoclonal antibodies, or we're working on peptides now for delivery. And there are several biological barriers, endothelial, cancer cell membrane, and I didn't have time to speak about endosomal membrane and drug release through endosomes. So all these steps are taken, and because my talk was the last one, to summarize beautiful basic science and development in nanoimmunology, I wanted to demonstrate maximum for treatment and preparation for FDA. But all molecular mechanisms are, studied in detail, and we try to do our full understanding how everything works.
Thank you.
Okay, there's one other question in the chat. Volker, if you can open the chat for me. There's a question for Toby regarding glucose uptake. Toby, are you still with us?
Yes, sure.
The question by Lorena Consolino was, did you also evaluate glucose uptake changes in your ISA-deficient mice?
Yeah. In the macrophages itself, we looked for that. So that's a bit tricky because the repolarization of macrophages also involves, if you like, a switch from more glycolytic activity to more oxidative phosphorylation. We don't have, based on the gene expression analysis of ISA-deficient macrophages, we don't have evidence that this is a general mechanism, so that ISA regulates mainly genes involved in glycolysis. But we would rather see this as a secondary effect, that we also see a metabolic switch in the macrophages.
Okay, thank you, Toby. I guess that all the remaining questions are related to Volker's talk on the protein corona.
Yeah. So I sparked quite a lot of controversies, it looks like, yeah. Especially when we complement, or when we looked at most of the people probably listening to the stream were also before with Zehntel, with Professor Zehntel, with Rudolf Zehntel and Professor Zehntel's talk. But I start with something else. What's the possibility of attaching a protein, an antibody of interest? Dr. Sebastian is asking. And basically, well, what we look mostly is, as proteins, is mostly antibodies, but we also were attaching interleukin-2. I think it's large enough, and if you can unfold something of the protein, then it's good that you that you have a chance also by pH modification, you can attach that. Johannes Kockelmann has been asking, what makes clustering bind to the particles? What's that? Well, we were looking if we can find a motive inside of clustering, and by using peptides. There is the peptide binding is not so well, is not so significant, but we find some stretches of peptides which bind to clustering peptides to PEG chains. So that's something. There seems to be a motive that is obviously recognizing PEG chains as a binding partner. And then there's a question from Anja Jakob. What factor must be considered with while attaching different antibody isotypes? That's a very good question, because instead, really, the antibody isotypes makes a big difference. We used IgG2A. That's answering a question from somebody else, actually, as well. And we saw if you use IgG1, for example, mouse IgG, in most cases we used here, then this is different. So the subtype is different, definitely, because you have a different FC part, is definitely something that is making a big influence. And that is dependent then on pH, which is optimal for that. And then comes to that question from Natalie and that Rudolf Ziendl showed that AF4 with the AF4 protein corona and self-populated nanoparticles such as liposomes is not detectable. Have you observed this also? What were the nanoparticles you were working with in your work showed? Were they hard nanoparticles? Yes, they were. They were basically all hard nanoparticles. As you can say, hard. We also have used nanocapsules, which are pretty hard still compared to liposomes. So there may be an influence of the underlying material. I think that's also what we see. We have used liposomes themselves, which were not pegulated. There you find definitely also something, a protein corona, and you find detectable amounts. But for these, other the systems as described by Rudolf Ziendl and colleagues, they did not find a lot. This goes also into this, what should be the effect of P-star shielding instead of PEG in terms of self-effect? We have not ourselves investigated that, so that question has really to be answered by Rudolf. We have not combined the two, so to speak, a polystyrene particle and then the P-star shielding. I think that would be also a good shielding effect with this P-star. And there was this other question about it, if softness makes a difference, and I think that that's really the case, that softness makes difference. It's also what the difference between what Professor Ziendl showed and what I show is that, there was another talk in this meeting in the cleanup sessions where they also used AF4, and they also found not many proteins on the surface. They also said they found basically no proteins on the surface. And I would suspect that actually the washing procedure is very much dependent on your analytical workup, what you find then afterwards. And I think if you wash with AF4, we wash so much that we wash away a lot of the proteins that we really have on the surface, and that a lot of people have shown also by other techniques. It's like Moing showed before with TEM images. So this is also something we have done, and now also other people have done, that you really can also visualize these. And I think if you wash very, very, very well, then you really get off most of that stuff, which is of course not a situation where what we have in the body afterwards, because there you have 100% serum or 100%. And actually, we should talk even how we can analyze while the other proteins are still around.
So I guess these were most of the questions that we have so far. this would conclude the session here. We will still have the option to go into a chat room number 1.
Right.
But for now, I think we conclude the session here, and we will reconvene into the chat room then in a minute, or you can use the break as you like. Thank you, everybody, especially for the presenters for their nice presentation, Volker for co-chairing the session, and have a nice rest of the conference. Thank you very much.